Thank you for that lovely introduction. I've got 5 minutes, so we'll jump right in. So, it's a question of success in sea lice biology is really what we're attempting to answer here. So you've heard a lot in the last session about biophysical models, and those are used in a variety of countries to make treatment decisions and various legislation, like in Norway, and as I said. And very little is known about the infection success of copepodids at different current velocities, and this is an element that's lacking in a lot of those models. And so, this is a question that I aim to answer in this pilot trial that I conducted last fall. And this has implications for treatment and modeling, which is the larger part of my master's thesis. You can see a model of Cobscook Bay in Maine there on the screen showing current velocity. So we'll sort of go quickly through just my salmon care, my prep methods. Atlantic salmon pulse molts were transported to the facility I did this at. They were kept at 35 salinity, 30, 10 centigrade, that feed. And then we settled on a flume velocity, so this was done within a flume, of 206 millimeters per second, and this was chosen because this was the slowest speed at which the flume pump could operate, and is also about the low to mid-level range of the current that sea lice in salmon and net pens experience. And gravid females were collected from naturally infected fish on salmon farms and then transported to the lab and reared to the infective stage. Let's see, sometimes this, there we go. Our methods, our execution of study. So, once they reached the appropriate number of copepodids, these were transported to the lab and counted. They were then kept in a glass jar inside the flume to maintain the same temperature as the flume during the duration of the trial. Copepodids were measured from that jar for an estimated 100 per fish. And these were done for 3 to 4 fish per trial. 43 were used in total, so there was just one that was used with 4 because 43 is not divisible by 3. And the copepodids were placed at the start of the flume, and this was done for a single pass. So based on my calculations, at the duration of, or at the speed and the duration of the flume, that was approximately 30 seconds. And then they were left in that flume for that point, at which time they were removed and then placed in a tank to recover. And so the lice were allowed to develop on the fish for 7 to 9 days. The fish were then euthanized with MS222 and died with neutral red, counted with a dissecting microscope, and the percent settlement was calculated for each fish. The mean settlement intensity, abundance, prevalence were calculated, and the total number of calamus that successfully settled over all of the fish throughout the experiment were counted over the number of copepodids that were estimated to be used. And our results. So in total, 12 fish out of 43 had lice, so this represents a prevalence of roughly 0.279. Our mean abundance was about 0.3 lice per fish. Our mean intensity was a little over 1 lice per fish. And 13 calamus were found over 4,650 copepodids that were used. And this gives us an infectivity rate of 0.280%. And our discussion. So our infectivity rate has implications for modeling, which again is the larger portion of the part of what this project goes into. If we know that an infectivity rate of 0.28 about, that means that we need roughly 280 infective particles in a given area per salmon to result in 1 successfully attached calamus. Little variance in my count data shows promise in my copepodid estimation methods. And there was no evidence of a double pass, as in lice were not able to survive circulating through the flume and attaching to the salmon. However, it was a lower abundance than what I would expect, and I think that this possibly could have been that I was so concerned about double pass confounding the results that I possibly knocked off some salmon with the net at 30 seconds because they didn't have enough time to attach. And this can be fixed in the future by increasing the challenge time that they're left in after. And more velocities will be tested this summer to compare and hopefully later incorporate in our model. And briefly, I'd like to thank the folks that helped get me here, my wonderful undergraduate crew, my advisor, Ian Bricknell, Kelly Cole, Damian Brady, several others, the folks at the National Coldwater Marine Aquaculture Center at the United States Department of Agriculture, and many others that do not fit on this slide. And I thank you for your time. If you have any questions, you can ask those at the Q&A, or you can find me at poster number 2. Thank you.